Welcome to the podcast, Dr. Helene Longeva. It's
Thank
truly
you
an
very much.
honor to have you with us today. I have so many questions for you and exciting topics to explore with you, so we'll just dive right in.
Thank you very much for having me.
Yeah. So, in Ayurveda, we talk about this concept called swasth, which means to be established in the self. In Ayurveda, health is seen as a dynamic balance of the body, of the mind, and the consciousness. And as in you're in harmony with your own nature, your prakriti, and you'll be naturally healthy that way. In other words, the emphasis is not on treating the disease, but on strengthening the system's innate intelligence so that there is no disease. The disease doesn't develop. And I think this concept resonates with salutogenesis, which I heard you talk about in one of your podcasts. So, from your perspective as a physician scientist, what do these concepts mean to you? Um, and how it might inspire a shift in how we think about health?
Yes, well, thank you. No, I think these concepts, these ancient concepts that have been around for centuries or even long, millennia, are very much resonant with what we talk about in terms of whole person health. First and foremost, it's really as two components. Whole person health as two components. One is the whole person. It's really not considering each organ or system in the body as being separate, but they're really part of a whole. And then the health part, which is related to the concept of salutogenesis that you mentioned, is the idea that uh, health and disease are not separate states, that they're really on a continuum and with opportunities for restoring health, or a person who can move from a state of being healthier to less healthy to finally develop a disease. But there are many opportunities along the way to reverse that and increase and build on the health of the individual. And everybody is unique in this way. Everybody has their own path and through life, but that the understanding, thinking about health this way is very resonant with concepts of traditional practices such as Ayurveda.
Yes. Um, so now let's introduce the audience to one of your favorite topics, which is the connective tissue. Uh, People have often thought of connective tissue as a structural scaffold, but some of your pioneering work and of others have shown that connective tissue is much more than that. So, for our listeners who might not be familiar with what connective tissue is and what fascia is, could you explain in simple terms what they are and about their functions in the body?
Yes, well, connective tissue is fascinating for so many reasons. One is, as you mentioned, it is a scaffold. It really is the what connects every part of the body with every other part. And without connective tissue, our body would just not be whole, right? And the connective tissue is surrounds everything, permeates all the organs, all of the visceral organs, but also the musculoskeletal plays a very important role in musculoskeletal system, and importantly connects the musculoskeletal system with the viscera, which is, I think, an aspect that is not very well understood in conventional medicine. But um, I became very interested in connective tissue because of my research on acupuncture and the idea of the what we call the acupuncture meridians, which is our sort of channel. They talk about them as channels going through the body that connects everything. It is very, very similar to the concept of connective tissue. So connective tissue plays a role um, that certainly a biomechanical role, there's no question about it, but also may play other roles in terms of communication throughout the body, perhaps also via the flow of interstitial fluid through the connective tissue, in addition to lymphatics and veins. There may be a lot of functions of connective tissue that we are just beginning to explore in research. So it's a very exciting field.
Yes, I agree. Um, it's a very exciting field, and people have to understand disease or health from a connective tissue perspective. That's even more interesting.
Yeah.
And I'll come to some of those questions later on. But my next question is, about some of the studies, some of the epidemiological studies that suggest that you had mentioned acupuncture, physical activity, and yoga, and they've all been mentioned in traditional medicine, can improve the quality of life, but the precise mechanisms still remain very less clear. So, how does connective tissue respond to these activities or practices, and why do you think it is important now more than ever to build rigorous scientific research and evidence around such modalities?
Yeah. Well, I wish we knew a lot more about that than we know now. And one of the reasons is that we don't have very good methods to study connective tissue in people in vivo, but that's changing.
Mm-hmm.
So up until recently, if you wanted to sort of visualize connective tissue in the body, you were pretty much out of luck because if you did, for example, radiology, for example, X-rays, CT scans, MRI, they're not designed to look at what we call the fascia or the loose connective tissue that is that network thing. They're very good at looking at bones, cartilage, intervertebral discs, these kind of harder structures. But this what they call soft tissues are not resolved very well at all. And so for that reason, diagnostically, we don't really have good ways to tell whether the connective tissue is healthy or not. One ways that people have used is their hands to palpate, and typically sort of what we call complementary therapies, massage, techniques, for example, that are part of traditional practices in both Ayurveda as well as like traditional Chinese medicine. They all have some kind of form of manual therapy, but also movement-based therapies like yoga. Or Tai Chi, for example, where people are instructed to learn how to move a specific way, and stretching, for example, gentle stretching can improve the health. But we don't have really good ways in science to measure that. And so just very recently, in the last 5 years or so, at the NIH, we have now a program to develop, as part of the HEAL initiative to address chronic pain, program to develop biomarkers and measuring tools to measure connective tissue, and to look at, they're focusing more on pain for understanding what we could talk about in terms of what's called myofascial pain. But I think that when these, I'll give some examples of these tools, but I think in the future that these tools will be very interesting and can be applied to other types of pathologies where connective tissue could play a part. So techniques, for example, like elastography, to look at not only the structure of the connective tissue, but also the mechanical stiffness, for example, of the tissues. Look at that in vivo, which is really difficult, but there's techniques now to do that. MR-based techniques, magnetic resonance imaging based techniques that can also look at, like MRS, elastography can be done with either ultrasound or MRI. Also different forms of MR spectroscopy, different forms of other types of very, very fine needles that can use to perform microdialysis in tissue. So many, many different techniques that are going to be developed as part of the program that can help us understand what types of pathologies are present in connective tissue and how can we help treat them and prevent them.
Yes, that's interesting. Some of the imaging modalities that I know of, breast cancer, where they image how the collagen is and how the architecture and alignment of the collagen and the stiffness.
Yeah.
So let's shift our focus to understanding cancer from a connective tissue perspective. This has always been one of my favorite topics and something that's really changed the way we look at cancer. For so long, cancer has just been considered a genetic disease, a set of mutations that would make cells grow uncontrollably. But when you look closely, we all know that's not the case. It's not just a ball of homogeneous cells, but it's a living ecosystem. It's more like a distorted organ. It's surrounded by extracellular matrix, fibroblasts, immune cells, nerve cells, blood vessels, and all kinds of chemical signaling that is happening, and the cells and everything is interacting within that ecosystem. And in fact, in some of the solid cancers like breast cancer, which you and I are researching on, or pancreatic cancer, the dense matrix is a big challenge. It can almost be compared to a bad soil. And thanks for mentioning that, the stiffness aspect. So one thing that stands out when surgeons look at these tumors are that they're very stiff. And it is now known that the tumors are about 10 times more stiff than a normal organ. And interestingly, Ayurveda also describes abnormal growths, which are referred to as granthi or arbuda, as being very stiff and immobile, very rigid. And the stiffness isn't just something that you can feel by touch, but like you had mentioned, there are imaging tools like mammograms or elastography that can actually pick up on these dense and stiff areas. And in fact, researchers have identified something called the tumor-associated collagen signatures, tags in the breast, which then can describe how collagen fibers realign and stiffen around the tumors, and they can also predict the aggressiveness of a tumor. So this brings me to a very interesting and emerging field called mechanobiology. And since there is stiffness around the tumor cells, how do they respond? Because the cells are always sensing their environment. So how do they respond to the stiffness, and what happens after that? So could you explain mechanobiology to our audience on a microscopic level? How do cells sense and respond to these mechanical cues?
Sure. Well, this is a topic that really has been a little bit neglected in medicine for a while. We were really, uh, throughout most of the 20th century, medicine was very much concerned about biochemistry, but without the sort of accompanying tissue forces and architecture that kind of where the biochemistry actually lives, right? It's not just molecules floating around. There's a structure there, and there are mechanical forces acting on them, and we're starting to catch up with that in understanding. This is why mechanobiology is so important. It's understanding biochemical phenomena in a structural and mechanical environment. So one of the fundamental sort of very simple equations in mechanobiology is a relationship between stress, strain, and stiffness, right? And the most important, the easiest way to explain to understand that is imagine you have two different rubber bands, and one of them is really stiff, and the other one is more sort of soft and pliable. If you hang the same weight from the two rubber bands, the stiff one will stretch less, and the soft one will stretch more. And so you can calculate the stiffness by if you apply the same force that like for example, hanging a weight, uh, and then you divide by you look at the force divided by the deformation, you can calculate the stiffness. And that gives you an indication of how stiff the rubber band is. It's the same thing in tissues. If you can either apply a known force and measure the displacement and calculate the stiffness, or apply a known displacement and measure the force. In practice, in tissues, it's usually easier to measure the displacement. It's called the strain. In the tissues, we have different kind of methods and techniques that you can measure the strain. We already talked about elastography, but in very small, at a smaller scale, there's something called atomic force microscopy, where you indent the tissues very, very fine, and then you can measure the amount of displacement that occurs and calculate a stiffness. So we can do this in tissues. These are not easy measurements to make, but they're doable. And people have started to look at different sort of biological phenomena by putting the mechanical forces back in and understanding if you change the force or the stiffness of the tissues, how does that affect the strain? For example, when a tissue becomes fibrotic, applying the same mechanical force will result in a lower amount of displacement. So you can start to do this kind of research. One of the things that has been surprising in the work that I've done in my lab is that we have found that the tissues, applying a certain force to the tissues can actually cause the tissues, the cancer growth to slow down,
Mm.
which is not intuitive, because we usually think that applying a mechanical force would then induce the tissues to sort of become more stiff, but that doesn't seem to be always the case.
Mhm.
So there are complex biochemical pathways that control the relationship between an applied force and sort of the remodeling of the matrix, for example, which we are just beginning to understand. So it's an interesting topic, very topic that we're just starting to learn how to explore.
I feel like to understand this better, we can shift our focus from tumors to something more relatable, something that we all do, that is stretching in a healthy tissue. So personally, after sitting for long hours at work, or after an intense workout or strength training or dance session, I feel like I feel so much better when I stretch. Um, Recently, I was having stiffness in my muscles, and my gym trainer told me that when the muscles feel stiff, it is because they are tight and contracted and almost stuck in a shortened state. But stretching helps them bring back to their natural length and releases that tension. So, from a scientific point of view, is that feeling of relief because stretching actually reduces the baseline stiffness in our tissues? Um, and stiffness is the same concept as you described, the resistance to deformation or stress and strain that we talk about in mechanobiology and mechanical engineering. And I imagine that the connective tissue behaves differently than muscle since it doesn't have myosin fibers. It's mostly collagen fibers, which have their own tensile strength. And as you pointed out, it's not very easy to understand how these mechanical forces travel across lengths. For example, if I stretch my leg right now, how would a cell below sense that stretch, and how would it react to it? So this is a very challenging things to study and a very challenging concept to know, but I would still like to hear your thoughts on
Yeah.
this.
So there are a lot of very interesting ideas that you sort of put together here. One is the fact that the type of research that we've done in my lab, we focused on the loose connective tissue, which is the connective tissue that is in between the layers of fascia. And the reason why this is important is because when we stretch, a lot of the movement that we do occurs what we call in shear, which means that you have all of the different organs are organized in layers, and you have different areas, layers that are more stiff, uh, separated by areas that are layers that are less stiff, and that's where the movement occurs.
Mm.
And what happens sometimes, and is that these layers are formed of loose connective tissue, which has collagen and has fibroblasts, but it also importantly has a lot of glycosaminoglycans or hyaluronic acid, for example. These are molecules that have a lot of sugar molecules, sugar moieties on them that bind a lot of water. And what happens is these glycosaminoglycans can be more or less sort of clumped together in such a way that the stiffness and the lubrication of these layers can be changed. And so a lot of times I think that when we stretch, when we exercise, like you mentioned, and we feel better, I think that what the types of movement that people are able to see are these shear plane movements in between that can change quite nicely as you engage the tissues and make and encourage them to move. Now, what does this have to do with the muscles is something that we don't yet understand very well, because there's a complex relationship between the connective tissues around the muscles and the muscle fibers. And so sometimes when we stretch, the connective tissue may engage and stretch a little bit more, which would then, what effect that has or doesn't have on the muscle next to it is still something that we need to understand. And this is where this whole techniques on imaging, for example, in myofascial tissues is going to be so helpful, is to help us understand the relationship between how the muscle is responding to the stretching and the increased mobility of the connective tissue around it. So again, a lot more questions than answers at the moment, but these are all really, really important things for us to understand if we want to develop techniques that help us stretch the tissues in ways that are beneficial. Because importantly, if you stretch too much, you can injure the connective tissue. So we have to understand what is the right amount of stretching that will promote relaxation of the muscle and reduce tension and pain. So these are all really important areas. And it's important in cancer as well, because what we have found in our work on cancer is that this different amounts of stretching can either increase the growth of the cancer or reduce it. So this is important. The dose matters. If you apply a mechanical stretching, and we can do this passively in an animal, in an anesthetized animal, if you apply a too large a force, the cancer grows faster. But if you apply too little force, the cancer also grows faster. But if you apply the force that is just the right amount, the cancer growth is reduced. So it's a very interesting and I think important area that we have to understand, because we want to be able to, if we want to use stretching as a way to reduce, for example, prevent the growth of cancer in tissues, for example, in the breast cancer, that's what we're finding in our animal experiments, is that stretching reduces the growth of these very early tumors. We have to apply the right amount of stretching so that we don't increase it, right? So before we translate this into applications in humans, we have to understand these dose responses very well.